Hello, I'm Dr. Charles Knight, and I'm from the Cal Poly Bailey College of Science and Mathematics. I work in the Department of Biological Sciences, and I'm a professor here of evolution and plant physiology and other disciplines. And I'm recording this series of lectures that I'm calling 10 Lectures on Evolution, which are kind of polished versions of some of the lectures that I give in my classes, in my class, Bio 414 Evolution. So enjoy. There's a channel, there's a number of these lectures. Now we're up to, this is number 6. So today, eventually, I'm going to be focusing on this target paper number 6 for our class. And it was published in 2024 in the Proceedings of the National Academy of Sciences, that's PNAS. And the title's Local Adaptation, Plasticity, and Evolved Resistance to Hypoxic Cold Stress in High-Altitude Deer Mice. Again, it was published in 2024, so it's a recent paper. It's made a splash in our physiology classes. It turns out there was another professor here, Christy Strand, who is using this paper in her upper division animal physiology class, and I also picked it up in my evolution class, and it's really a cool experimental design, and we'll get to this paper eventually. But before we do that, I want to talk a little bit about Jay Stortz, the primary PI involved on this paper. Of course, there's many other people, and I'm going to only focus on Jay Stortz primarily because he's going to be a thread that goes through a number of other kind of supporting papers that I'm going to introduce in this lecture too. So Jay Stortz is there, and I just want to remind my class, and then also people that have been familiar with this channel, that we already talked about a paper by Jay Stortz, which was about birds and high elevation adaptation in birds, and this is beautiful paired comparison design. All the blue rows, or the darker blue, are high elevation birds paired with a lower elevation bird, and they sequenced all the hemoglobins and looked for parallel substitutions, and only rarely found that, but they did at position 83. Anyway, I don't want to go into that whole story because that's what we did in the last lecture. But today we're going to talk more about another cleverly designed experiment on kind of plasticity and acclimation, and also in the background testing high and low land mice. And I think both of these papers are really cool from the experimental design standpoint, and I like to emphasize to my students, if your design is unique but also rigorous, it really doesn't matter what you find, because everybody will be interested in the results. And I find the work of Jay Stortz to be very inspirational in that regard. A great experimental design, interesting results. They're kind of complicated, but that's the messiness of science, and we're going to get into that today. So Jay Stortz works at the University of Nebraska at Lincoln. He got his BA at the University of Colorado, and also a PhD at Boston University, and then now is back at the University of Nebraska, and has really contributed a lot to animal physiology and evolution and plasticity, especially revolving around hemoglobin. And I think he's really carrying the torch of Max Pruitt's. I like how in many of his papers he cites Max Pruitt's really early on. And the data that he's finding don't necessarily go against what Max Pruitt found. Of course, Max Pruitt's won the Nobel Prize on hemoglobin, but wow, the incredible amount of detail and really creative experiments are inspirational coming out of Jay Stortz's lab. I also like to mention that Jay Stortz has an army of collaborators, both in North America and also in South America. The first papers we're going to look at today deal with volcanoes in South America. We've got a bunch of collaborators like Naim Batista and others from Chile and Argentina, and maybe that's Colombia, and other investigators here in North America. So an incredible collaborative team that has produced all of these papers, and I'm just highlighting a few of them here. But I would spend a lot of time going into each one of the authors if I did that. So we got to move on, because I want to finish this in less than an hour. That's my goal. So the backstory here, I'm going to talk about a paper that led up to this paper that was published in 2022, and it describes some high elevation mice that Jay Stortz and these collaborators found at very high elevation. There have been records before, and they really wanted to document these mice because it's a hemoglobin lab, and they were like, maybe if we sequence the hemoglobin, we'll see a dramatic difference or a base pair mutation or amino acid change that explains how these mice can live at such high elevation. And this is Mount Lilienko, and there's mice all the way up at the top of that mountain at 22,000 feet. And that's incredible. And this mountain is unique because it's right at the edge, or maybe even considered inside the Atacama Desert. There's very little snow up there, much less snow than you'd expect in North America for a peak that's that high. Remember, it's the Humboldt Current that creates the Atacama Desert, that cold water current washing the coast of Chile, and creates a very dry environment all around here. So it's a unique snow-capped peak in that there's not much snow up there. But this little mouse, the Andean leaf-eared mouse, lives all the way up there at the top. And this paper actually is first just documenting that it occurs there, and then sequencing the cytochrome B gene from this population and other populations across the Andean chain, and also comparing them to lowland mice. So just a quote from the paper suggests that because the upper mountain is characterized by low atmospheric pressure, extreme temperature fluctuations, extreme aridity, intense UV radiation, and a total absence of vascular plants, it is considered one of Earth's closest analogs to Mars. And so I think that this is a great example of, I don't know, Jay, did you get money from the NASA Astrobiology Institute to do this work? Because it'd be a great use of it. Bioprospecting for animals up there at the top of this mountain. They looked in soil samples too. They found 13 live and mummified mice from various Andean peaks above 20,000 feet. And they also sampled in soil on runways, when kangaroo rats around here make these paths and they have the common trackway where they normally run. They scooped up the soil in those runways and did soil DNA analysis looking for other organisms, and they found a bunch. I'm not going to go into those details. Mostly I'm interested in the hemoglobin from the high elevation and low elevation, and also how these mice are related. So they did find evidence of mice and other plants, animals, and microbes, of course, from these soil samples in runways. So this is the paper that they published that described these results, observational studies that involve DNA sequencing of these mice from high elevation. They sequenced the cytochrome B gene from these 13 populations, published it in Journal of Mammalogy in 2022. And I'm calling this the backstory because the other papers we're going to deal with are broader, deal with more different species, or involve more detailed physiological adaptation or acclimation studies. So these are the sites. These are the volcanoes across the Andean Cordillera, or is that how you say it? Just the Andean chain of mountains. Notice there's 13, and each dot represents a different collection location for a high elevation mouse. And also population 1 and 2 are from sea level, so they had those for comparison. Number 5 is the Mount Lilienko that I showed you pictures of already. So they're close by to 1 and 2. So they've got pictures of mice actually living up there, and they've got mummified mice, mice that died up there. So it's pretty much indisputable that mice are living up there somehow. So this lab, Jay Stortz's lab that deals with hemoglobin, is really curious. What is their blood like? How can they do that? So they did this detailed physiology work that starts to illuminate that. They used a bunch of degenerate primers. The degenerate primers use the IUPAC code for base pair positions, and I'm not going to go too much into detail on that here. But this list of primers, there's a star if it was successful, that is, it amplified a piece of DNA, and the primer's on the right. So that really there's a list of primers that you could use if you were studying mice at high elevations and you wanted to know something about cytochrome B, not cytochrome C, cytochrome B. That's the one they studied. And let me just look at this primer sequence in the IU. All right, I just wanted to remind you what this degeneracy of the primer code means in the IUPAC table. The IUPAC code shows how there's a different lettering code that's different than A, C, T, and G. Those are the standard base pairs, but then R, Y, for example, R is A or a G, Y is a C or a T. It goes all the way down to M, which is an A or a C. If we look at the first primer sequence, it's CCM, T, C, C, A, A, C, A, T, Y. So the M and the Y are the degenerate sites. M would be an A or a C, and Y would be a C or a T. We've seen these in signature sequence signature sequences for Hox genes, where there's some degeneracy in those signature sequences. Likewise, when studying primers here for cytochrome B, they use degenerate primers just with the hope of finding a primer that worked, that amplified a section of DNA across a number of different species or a number of different populations that then they could do a phylogeny for. So that's why they did that. Glossary notes here: a degenerate primer is a collection of similar PCR primers that can amplify a variety of related DNA sequences. The IUPAC code is the International Union of Pure and Applied Chemistry, and it's just a nomenclature for DNA identity, I guess you could say, or composition. Cytochrome B is a conserved protein that is part of the respiratory chain complex 3 in mitochondria. A polytomy, which will become important soon, is a branch in a phylogeny with many taxa branching off of it. And we're going to see a polytomy in the phylogeny that they created. These are the 13 locations across the Andean chain, the peak of volcanoes. The Lilienko is number 5, the red star. And they compared those high elevation peaks to 2 low elevation sites, number 1 and 2. This is the phylogeny that they produced, and it's hard to see, I know. But at the end of each taxa is a number, and those numbers correspond to the peak. And I'm just going to focus on one little part of this phylogeny because it really tells part of the story that's interesting, I think. So we're going to look at just this part. When we blow that up, you can see that 5 are Mount Lilienko sequences, which are spread throughout this upper branch of the phylogeny. You can see that they're nesting really closely with populations 1 and 2, which are close by-ish, probably 100 miles away as the crow flies, maybe further. I suppose the Chilean biologists were like, you idiot, that's much further than that. But it's relatively close compared to other volcanoes. So 5 and 1 are on a polytomy in the phylogeny, basically identical cytochrome B sequences for the primers that they were able to amplify. This is interesting to me because it's a mitochondrial gene, which leaves open the possibility that there's been some introgression of the mitochondrial genome from one population to another population. That's a whole nother topic, but it's possible. But regardless, for the mitochondria, we see that the mitochondria might be fairly similar, and if there's this mitochondria, if it is the same mitochondria or introgressed, is functioning at sea level as well as high elevation. So that's remarkable in itself, I would say. That's kind of a sidelight. But that's kind of one of the main take-homes of this paper, at least for me. They barely found any differences in cytochrome B between some of their high elevation and low elevation mice, and they quoted that it's as if there were the same large, widely distributed population. And when I think about it, It's as if these mice just walked up to 22,000 feet and decided just to live there. And that's pretty remarkable. How can they do that is the question, I think, of this paper and the rest of, or a lot of Jay Stortz's other papers that I'm going to present today. And there's only going to be two more, and one of them is our target paper. So this was published in 2022. I'll just remind you, a fairly recent story, and Jay Stortz was one of the investigators. Now I'm going to go all the way back to 2010 and a paper that Jay Stortz published in the Journal of Experimental Biology with the coolest cover ever, I would say. Here we've got mice with the hemoglobin molecule. You can even see the heme group sticking in there, the chemical, which is actually kind of the active site of hemoglobin. That's a beautiful cover. Kudos to whoever made that. But this is going all the way back to 2010, quite a while ago compared to today, but it's still published by Jay Stortz and another series of investigators. And in this paper, the title is Genetic Differences in Hemoglobin Function Between Highland and Lowland Deer Mice. And for this, they're studying something a little bit closer to home, Mount Blue Sky in Colorado, that's at 14,000 feet, and mice from Kearney, Nebraska, 2,100 feet. So about a 10,000 foot difference between populations of deer mice on Mount Blue Sky and at Kearney, Nebraska. Mount Blue Sky is the formerly known Mount Evans, which is on the Front Range scene. If you're in Denver, the highest peak you see right outside of Denver is Mount Evans. And the name was changed last year in 2023 because Governor Evans, back in the 1800s, was the governor who participated or ordered or at least was complicit in the Sand Creek massacre of Indians. So there's been a push to rename these mountains for a long time, and it finally happened last year. Glossary notes here. This paper is going to involve allostery a lot, and I think it's important, and the rest of this presentation involves the concept of allostery, so I thought it'd be important to define this before we even even see it. I know I'm not even looking at it yet because it's a detailed chemical principle, but it's very important to understand the rest of these papers, so let's try. An allosteric site is a site in a protein, and that is an amino acid position in a protein that interacts with other proteins or chemicals that alter the active site. That's the definition of allostery or an allosteric site. For blood chemistry, the pH often can be modified by adding KCl and DPG, which is diphosphoglycerate. They both affect the oxygen binding, and we would say that pH and DPG affect oxygen binding allosterically. That is, they're chemicals that, and other, they're both chemicals, or the pH is a condition that affect oxygen binding, not by an amino acid mutation at the active site, but rather changing the environment that changes the affinity of oxygen for the heme group. DPG, diphosphoglycerate, is a metabolite that's normally found in red blood cells. So this is something that organisms make, and it's normally present in these red blood cells. Blood pH can change with increased respiration, and so you should know, or I'll just tell you that when CO2 is produced by respiration, it can dissolve in the blood and become carbonic acid, H2CO3, which would decrease the acidity or decrease the pH or make it more acidic. So organisms constantly face this change in the blood environment. If the goal is to deliver more oxygen to the heart, let's say, changes in pH of the blood or changes in DPH can modulate the oxygen carrying capacity of the blood. And it may turn out that mechanisms that change blood pH or DPG or other allosteric chemicals in the blood may be more important in adaptation to high elevation than changes in hemoglobin amino acid sequences. That's at least part of my conclusion going forward about this group of studies. and we'll get back to that in a minute. So this is the paper again. I'm calling this target paper 6B. It's not our target paper 6, but a supporting kind of backstory paper. Again, title is Genetic Differences in Hemoglobin Function Between Highland and Lowland Deer Mice. This is their chart, and P50 is on the y-axis. P50 is that parameter that we saw previously in the target paper 5, and that's similar to the Michaelis-Menten kinetics, the oxygen concentration at which 50% of the hemoglobin heme groups are bound to oxygen. They're going to study this in a number of different conditions. Stripped means it's basically just hemoglobin in a buffer, I assume. I've never done this, so I don't know exactly what it looks like to have stripped hemoglobin, but I assume it's just blood without, well, certainly without DPG and KCl. So at that condition, stripped hemoglobin, comparing the high and low elevation mice, you'd expect the P50 to be lower in high elevation mice because it would take less oxygen to bind or to occupy 50% of the active sites, because these organisms would be trying to carry more oxygen at lower oxygen concentrations. So P50 is slightly lower, but I don't think that it's significantly different between the high elevation in black bar and the low elevation in the gray bar. So when they found this, they did all of this work. They went up to Mount Evans, Mount Blue Sky, collected those mice, compared it to these mice from low elevation in Nebraska, and I expect they were expecting to see a much starker difference between the high and low elevation P50s, but they didn't. When they added DPG, that didn't change the story. But when they changed the pH of the blood with KCl, that led to a significant difference. The high elevation mice had a lower P50 than the low elevation mice. So that is significant. And the combination of DPG and KCl, again, was not significantly different. So my conclusion from this is that allostery really mattered, especially blood pH, for P50. The pH affected how hemoglobin bound oxygen. That's going to be important going forward. If we go on to another paper that J. Stortz published in 2016 at the same time as that bird on mountain paper, the high elevation, low elevation comparison in birds. He titled this review paper of Hemoglobin Oxygen Affinity in High Altitude Vertebrates. Is there evidence for an adaptive trend? And I'm just going to give you the conclusion of this review, and I'm going to come back to this later and look at more of the data. But what J. Stortz says is that in mammals, the evidence for a positive relationship between hemoglobin oxygen affinity and native elevation is equivocal. Sometimes it is lower P50 in high elevation animals, sometimes it isn't, like we just saw. In birds, by contrast, there's a remarkably strong positive relationship between hemoglobin and oxygen affinity and native elevation. And then, in fact, the data for high altitude birds provide one of the most compelling examples of convergent biochemical adaptation in vertebrates. So I just underline that here. Pretty compelling story in birds. Remember, that came from this paper back in 2016. This is a review paper that was published on the same month, October 2016, as the cool science paper that we already studied. But this is all leading up to this new paper that was just published in 2024, last year. And this title, again, is Local Adaptation, Plasticity, and Evolved Resistant to Hypoxic Cold Stress in High Altitude Deer Mice. And again, that's published 2024. I'm going to give you my take-home points now. Usually I do this much earlier, but I had to give some of the backstory of even different papers that builds up to this paper. And now I'm going to give you what I think are 5 take-home points from this whole series of observations across all of these papers. And that is, first, that biochemical plasticity for oxygen delivery at high elevation is pervasive in birds and mammals, and that's delivery. It's not P50 necessarily, but the delivery of oxygen. Biochemical adaptation for increased hemoglobin oxygen hemoglobin binding at high elevation is pervasive in birds but equivocal in mammals, as they just said. Mice can live at some of the highest elevations possible, and they are closely related to sea level mice, and that's remarkable. Mice can acclimate to high elevations through various physiological means. Oxygen delivery and biochemical phenotype are important, and allostery is very important. I'll finish these take-home points with the really philosophical and important consideration that the traditional dogma of adaptation involves amino acid changes in proteins may only explain part of how animals can live at high elevation. Allostery, the chemical environment of the blood, may be very critical to consider too, but I'll just point out that these allosteric changes may be due to the traditional dogma of adaptation involving amino acid changes in proteins. So it's just that the candidate gene that we looked at, hemoglobin, at least in these deer mice, wasn't like that different. It really wasn't that different, or not as different as I expected it to be between high and low elevation mice. But if you change the blood pH, it is different. So I'm going to read the abstract of this new paper from 2024. This is just my tradition. I like to couch the presentation of a new paper with the abstract because the scientists have thought a lot about what to put in there. And who am I to blow past this? And it seems that it really helps me conceptualize the paper better if I ground it in the abstract. So here we go. A fundamental question in evolutionary biology concerns the relative contributions of phenotypic plasticity versus local adaptation in enabling wide-ranging species to inhabit diverse environmental conditions. Here, we conduct a long-term hypoxia acclimation experiment to assess the relative roles of local adaptation and plasticity in enabling highland and lowland deer mice to sustain aerobic thermogenesis at progressively increasing elevations. We assess the relative physiological performance capacities of high and lowland natives as they were exposed to progressive stepwise increases in hypoxia, simulating the gradual ascent from sea level to an elevation of 6,000 meters. The final elevation of 6,000 meters far exceeds the highest attainable elevations within the species range, and therefore tests the animal's ability to tolerate levels of hypoxia that surpass the prevailing conditions within their current distributional limits. Our results demonstrate that highland natives exhibit superior thermogenic capacities at the most severe levels of hypoxia, suggesting that the species' broad fundamental niche and its ability to inhabit such a broad range of elevational zones is attributable to genetically based local adaptation, including evolved changes in plasticity. Transcriptomic and physiological measurements identify evolved changes in the acclimation response to hypoxia that contribute to the enhanced thermogenic capacity of highland natives. That's a lot. That's a dense abstract, fairly long. I'm going to highlight some words and phrases that I think are important to kind of define for my students and any viewer. So I'm just circling them here, and I'll start in the glossary notes page explaining these. We'll start with phenotypic plasticity. That's the same genotype producing a different phenotype, and we can see that in blood chemistry. Local adaptation is adaptation to a local environment in the same species. Thermogenesis is the process of producing heat in the body, and we'll see this under chilling experiments. Acclimation is the physiological accommodation of novel environments, you could say. That certainly involves phenotypic plasticity, or acclimation does involve phenotypic plasticity. Hypoxia is just low oxygen concentration. The fundamental niche is a range of environments an organism could occupy, which is different than the realized niche, which is a range of environments that an organism currently does occupy. So this is their cool experiment. This is figure 1A from this PNAS paper. I'm going to take some time just to look at it. Let's start at the top. We have the experimental time, 7 weeks. We've got control groups and acclimated groups. We've got the little mice emojis. The lowland mouse is open, that is, it's white. The highland mouse is black. That's true for both the control and the acclimated groups. The control population is always kept at 21 degrees or 21% oxygen, normal barometric pressure oxygen. And the acclimated groups, well, so that was through the entire experiment. Every time they sampled at each one of those open boxes, it was at 21% in the control group. In the acclimated groups, they tested at 0, or they acclimated them to 0, 1,000, 2,000, 3,000, 4,000, 5,000, and 6,000 meters. And they didn't actually take the mice to those elevations, they just exposed them to the barometric oxygen concentration at those elevations. So they simulated those environs that way. Cool experimental design, and so we're curious what they found. Remember that they're studying mice from Mount Blue Sky and Kearney, Nebraska, and it's not 500 feet, that's 500 meters. It's more like 2,200 feet in elevation in Kearney, Nebraska. Comparing that to Mount Blue Sky, which is at 14,268 feet, and the mice actually are living there. They live in the snow, I guess, or under rocks on Mount Blue Sky, and just wherever mouse do down by the Platte River in Kearney, Nebraska. They actually did their physiological measurements in their control populations were reared at McMaster University, so not even at Lincoln, Nebraska. So this is quite an international collaboration. First, Jay Stortz was working down in South America with a bunch of South American collaborators, and also those collaborators working in his lab in Lincoln, Nebraska. But then a lot of this physiology was carried out at McMaster University. Measurements were made on mice born and reared in captivity from the field collected mice, so there were no maternal effects. 32 mice were used. The whole experiment was replicated twice, and I'm going to show you the replication here next. This is the diagram of the replication. It's pretty complicated, but all mice were tested independently at the PO2 of the acclimated or acclimating mice. So all mice were tested independently at the partial pressure of oxygen of the acclimating mice. So even the control mice were exposed to those different oxygen concentrations, and we'll look at how those are different between the ones that were acclimated to those elevations. Let me explain one of these groups. So this group, the control lowland mice, there's 4 males and 4 females, 8 mice total. In the graphs, that's going to be an open circle. Next, we have the highland mice in the control conditions. 4 males, 4 females. That's going to be the closed circle. Next, we have 4 males and 4 females of both the lowland and highland mice in the acclimated conditions, and those will be the open and closed triangle. Same scenario for 1,000 meters in the control, open circle, closed circle, in the acclimated, open triangle, closed triangle, 1,000 meters, 2,000 meters, 3,000 meters. And I just interlude here that they're measuring VO2 max, and VO2 max is measured in a respirometer with chilling and heliox using the PO2 at each acclimating elevation for all 4 groups of mice. All of them were exposed to the same PO2, and they were chilled. And I just talked to Dr. Blank, a physiologist in our department, and I'm like, why do they chill them? And he's like, well, the shivering thermogenesis is a more controlled way. Instead of trying to get these mice to run on a wheel, you just chill them and they shiver, and that's a high metabolic load, just like as if they were running on a wheel. So they used heliox. I think it's a controlled condition, a replicatable condition. So VO2 max is the highest amount of oxygen that an organism can use during intense exercise, and I should have put in there or chilling thermogenesis. It's basically the highest metabolic rate. Heliox is a mixture of helium and oxygen used to standardize conditions for VO2 max measurements. So that was an interlude. They did the same thing at 4,000, 5,000, and 6,000 feet. So clever design of the study, and now we just look at the data. It's going to be a complicated graph. It's going to have a lot of open circles and closed circles, open triangles and closed triangles. Just remember what those are. There's a legend in there. This is a big result. This is figure 1b on VO2 max. On the y-axis, this is the maximal O2 consumption relative to the highest value. So the peak value was a 1, and it's not even really shown here because it was one of the groups, probably the one at 2,000 meters of the high elevation control mice that used the most oxygen. But then it was average, so we don't see the highest value. The x-axis is elevation. I'm just going to go slow here just so you kind of get used to looking at this graph. Each circle and triangle corresponds to the legend, which is in the lower left-hand insert on the graph. The big comparisons we want to make is between the triangles and the circles of a similar acclimation class. For example, at 6,000 meters, we see that VO2 max was highest in the acclimated highlanders compared to the acclimated lowlanders. A pretty big difference there. Goes in the predicted direction. Same in the control environment though too. The control highlanders had a higher VO2 max than the control lowlanders. The open triangle was higher than the open circle. I don't know if that one was significant, but it looks like it might have been. In general, across this whole bunch of data, you see a decline. There's a decline in VO2 max going from low elevation to higher elevation. So there's some stress. We would call that stress in physiology. The mice aren't performing as well at high elevation. Not delivering as much oxygen. There's less oxygen consumption, or less maximal consumption. If we look at other points on this graph, we could go back to sea level. We see that the control acclimated, or the acclimated lowlanders, really the acclimated lowlanders, the closed circle and the open circle should be the same, and they're probably not significantly different, even though it's a little bit higher. But what we do see is the highlanders are a bit lower. Don't know if that's significant or not. Lower VO2 max for the highlanders at sea level. You could say, well, they're adapted to high elevation. They don't do as well when there's so much oxygen. If we go to the mid-range of latitudes like 2,000, we see that there's maybe beginning to be a reversal where the highland mice, well, I don't know, they're fairly similar. But then it's really at the higher elevations where, at least if you're following the acclimated highlanders, though, that group of mice tends to outperform, going from 3,000 to 4,000, actually from 4,000, 5,000 to 6,000. Those are the highest VO2 maxes at those elevations, the highland acclimated mice. But the control highlanders, well, they did better. Anyway, I'm kind of just narrating this figure. Statistically, all those groups were significantly different. They had enough replications to get there. So there was significant phenotypic plasticity, I would say, across the elevations. And there was also a genetic difference between the populations. In an ANOVA, that accounts for them being lower in some places and higher in other places, but there was a statistically significant difference. You might be curious what P50 looked like, and there were a lot of physiological data presented in this paper, and P50 was one of them, but then there were so many other parameters, and I only presented VO2 max and P50 because many of the other parameters were correlated with P50 or VO2 max. So let's just look at P50. P50 we'd expect to be lower for highland adapted mice, and we see that at 2,000 meters, well actually from 2,000, all the different elevations, the highland mice, whether they're control or acclimated, had lower P50 compared to the lowlanders, which are the circles. That's true at every elevation, and it's statistically significant. So P50 was lower in high elevation mice. All right, that leaves the results from the PNAS paper, but I'm going to place those results in the context of another one of the target papers I already introduced, and that's this one on hemoglobin oxygen affinity in high altitude vertebrates. Is there evidence for an adaptive trend? They've done a comparative study here. The gray outline taxa that's filled gray are high elevation taxa. The white lines are low elevation taxa. They've got marmots, pikas, mice, and pumas, big cats. So this is the kind of comparative study that I like. It's the reason why I chose the bird paper. This comparative study is very similar to the bird paper, and realize they were published in the same year, 2016. So let's take a look at the results for P50 in deer mice. P50 in deer mice, we just saw a little bit lower in the high elevation mice versus the low elevation mice, but not dramatically different. In pikas, we see maybe a slightly bigger difference. High elevation mice had a lower P50 than the low elevation mice. Or sorry, lower P50 than the low elevation mice. And now we go to the marmots. Opposite trend of what we predict. And the big cats, similarly opposite trend. Overall, the differences were not really that significant, and it might only be under allosteric conditions where we add KCl where the difference becomes significant in deer mice and pikas. But then looking at marmots, it still goes in the opposite direction. And big cats, equivocal, same, pretty much exactly the same. What about DPG? Same sort of trend, maybe significant, maybe not in deer mice. In pikas, a little bit more, but in marmots, it goes actually now in the predicted trend. But look at the KCl versus DPG in marmots, just flips around, and big cats, not anything really, no big difference. Last, KCl plus DPG, predicted trend in deer mice, predicted trend in pikas, opposite or not significantly different in marmots and panthers, sort of in the predictive trend. This is a lot of work to get all these comparisons, and they were not really what they expected to see, I would assume. So allostery probably matters a lot, but that's what they were able to show for hemoglobin P50s and VO2 max. So I'm going to go back to my take-home points from all of these studies. The biochemical plasticity for oxygen delivery at high elevation is pervasive in birds and mammals. So oxygen is probably delivered better, but the biochemical adaptation for increased hemoglobin, oxygen, hemoglobin binding at high elevation is pervasive in birds, but only equivocal in mammals. Sometimes it's there, sometimes it's not. Mice live at some of the highest elevations possible, and they are closely related to sea level mice. Mice can acclimate to high elevations through various physiological means that might involve oxygen delivery and the biochemical phenotype and allosteric interactions. And lastly, again, I just want you to really think about this last quote. The traditional dogma that adaptation involves amino acid changes in proteins may only explain part of how animals can live at high elevation. Allostery may be critical to consider too. However, but allosteric modifications may involve traditional biochemical adaptation. So it just suggests that maybe this candidate gene, hemoglobin, may not be the locus of adaptation for hypoxia stress. There could be other loci of adaptation, and I'm sure Jay Stortz is considering doing a QTL analysis in these high and low elevation mice, breed them together, get an F2, get a bunch of SNPs spread throughout the genome, see where those SNPs are and the association with the phenotype, and see where they pop up. It may be in the enzymes that produce DPG or the enzymes that affect blood pH. I'm not sure what those enzymes might be, but it seems like that would be a logical next step in this research. Thank you for tuning in to my 10 lectures on evolution series, and this was a early slide that I made that kind of encapsulated all of these papers, but it was way too busy, but I liked the pictures, so I left it in. Okay, bye-bye.